seminar. RCMI stands for Research Centers in Minority Institutions. My name is Bill Sutherland, and I'll be your moderator for the seminar presentation today. And I'd like to welcome you to our first seminar of this semester. And I'd like to start by saying for the last year, one thing that's been on everybody's mind has been COVID-19. And probably for the last 2 years, for the last 2 months, the other thing that's been on people's mind has been the vaccine. And to the point that when you hear someone say the word vaccine, you don't have to wonder what vaccine they're talking about nowadays. You know they're talking about the COVID vaccine, just the word vaccine. They're not talking about the flu vaccine. They're not talking about the shingles vaccine. When they say vaccine, they're talking about COVID vaccine. And so it's been probably the most anticipated and talked about vaccine in my lifetime that I can recall. So, and because of all that, I am very pleased that our speaker today is Dr. Bonnie Graham. He is the director of the Vaccine Research Center at the National Institute of Allergy and Infectious Diseases at the NIH. And he is the chief of the Viral Pathogenesis Laboratory. And he is also the lead investigator for the development of the Moderna mRNA COVID-19 vaccine that is currently being distributed under emergency use authorization. His primary interests are vaccine development for viral diseases, viral pathogenesis, mechanisms of immunity, and pandemic preparedness. And as director of the NIAID Vaccine Research Center, he directs basic laboratory research and has developed novel vaccines for the RSV, influenza, Zika, and as well as coronaviruses. His laboratory explores the structural basis for antibody-mediated viral neutralization. He investigates basic mechanisms by which T cells affect viral clearance and immunopathology. Dr. Graham has a BA from Rice University, an MD from the University of Kansas School of Medicine, and a PhD in microbiology and immunology from Vanderbilt University School of Medicine, where he also completed internal medicine residency, chief residencies, and a fellowship in infectious diseases. And today he's going to be talking to us about rapid development of COVID-19 vaccines. And before I turn it over to Dr. Graham, I ask that you hold your questions until the end of the seminar. And also, if you like, you can use the chat and put them in the chat during the seminar, and we'll be monitoring the chat, and we'll pull questions from the chat during the question answer period. And one final note, this seminar is being recorded, and at some later date, it will be loaded on the RCMI website for later viewing. And please remember to keep your devices muted. And now, without any further delay, it is my great pleasure to welcome to you Dr. Bonnie Graham. Welcome, Bonnie.
Thank you, Bill, for that kind introduction, and it's good to be with you today. It's always a privilege to visit Howard, I guess virtually this time, but I'm glad to be here. And I'm going to talk about the rapid COVID vaccine development, more about the early part of the story, and then we can maybe talk about what's happening right now near the end. I work at the Vaccine Research Center. This was a place founded in about 20 years ago to develop an HIV vaccine. And even though we have not been successful with that development, the work trying to make an HIV vaccine has generated a lot of new technologies that just didn't exist 20 or 30 years ago. And they have allowed us to approach and solve some of the unmet medical needs in these diseases, including emerging infections that I list here. We can do that because not only do we have this basic research building, but we have almost 100 engineers in Gaithersburg who do process development, and another 100 or so people doing pilot GMP manufacturing in Frederick. We have our own self-contained clinic at the NIH Clinical Center and a laboratory devoted to analysis of those clinical specimens. It's managed by this relatively small group of principal investigators and program heads. And over the years, we've developed different ways of delivering vaccines, including nucleic acids that I'll talk about today. We've also done vector-based approaches, either pox or adenovirus vectors, virus-like particles for chikungunya in particular, and protein-based vaccines. Bill mentioned the RSV, and these are some of the candidate flu vaccines that are now in clinical testing. We've also developed a number of antibodies, and we've sent these products around the world. And just as an example, one of the antibodies we made, the MAB114, was used in Eastern Congo in 2018 during that Ebola outbreak, and was found to effectively treat Ebola. In people infected with Ebola, we could reduce mortality to less than 10% by treating with this antibody. And that has now been approved as an Ebola treatment, and it's called Ebanga. So HIV and Ebola and other viruses listed here in the middle have been emerging since well over 40 years. Since the beginning of my career, we've seen the emergence of all these different types of diseases, and these are not going to stop coming. And so I want to talk a little bit about how to think about or organize a way of thinking about pandemic preparedness for these pandemic threats that we know will continue to come. They're either based on zoonotic transfer through an intermediate animal model, or vector-borne diseases, as we've seen with Zika or chikungunya. And even HIV, you can think of as an emerging zoonotic infectious disease. And at one time was a regional problem that, because it was unrecognized, became a global problem, which is going to be one of the themes. And in the past, up until now, all these emerging viruses and the need for improved licensed vaccines outweighed our capacity for what we know how to do, which is passive surveillance and contact tracing and quarantine. But over these last 20 years, these new technologies, we think, turns vaccine development or biologics development into more of an engineering exercise where you can have more concrete timelines and do better milestone planning. And so this list of new technologies, I won't mention, won't go over here, but there are a lot of things that we could not do 20 years ago. And as you think about the history of vaccine development, these first four, from smallpox up even to influenza in the 1940s, were really the work of individual people and efforts and champions trying to just get this vaccine through. But these other two clusters, one in the 50s and 60s and another one since 1980, have been the result of new technology advances. And cell culture allowed you to grow viruses in large quantities to either make live attenuated or whole inactivated virus vaccines. Molecular biology allowed you to make molecular clones or reassortants or recombinant proteins and things like hepatitis B or HPV vaccines that weren't possible before that technology. And we have thought, and I gave this seminar a few years ago at Howard, that maybe structural biology would be one of the new technologies that would allow us to have a vaccine based on structure-based design concepts. And the RSV vaccine is indeed advancing and may be a vaccine, but this other problem, coronavirus, snuck in before that, and it includes both structural biology, gene-based delivery, the availability of rapid gene synthesis and manufacturing. These technologies have made the coronavirus vaccine development during this last year feasible. So just as an example of what I think is the main technology that really has changed the way we think about vaccine development, and this is respiratory syncytial virus, which is a pneumovirus. It's an envelope virus, and it has a protein on its surface called F, or fusion protein, that is analogous to the spike protein of coronaviruses. And so it is a class 1 fusion protein that massively rearranges to mediate membrane fusion. And what we found was we were able to capture the shape of this protein in its original native functional conformation. And with that, found some new sites at the apex of this protein that are highly neutralization sensitive. And typically this protein rearranges and turns into something shaped like this, and this epitope that's so sensitive is lost. And over the last 30 to 40 years, people have done at least 5 or 6 different vaccine development programs with this molecule, and always gotten about the same result, which is a boosting of 2 to 3-fold of the original neutralizing activity. But if you can learn how to stabilize it, as we did with this C-terminal trimerization domain and internal disulfide and cavity filling mutations, and maintain this shape, use that as a vaccine product, we now know, and it was published just last year, that this protein can now boost neutralizing activity by up to 16-fold or more, which now makes it a candidate for a maternal immunization that could perhaps protect babies through 6 months of age. And just to make it clear how much these proteins change over time and how important it is to capture this shape right here of the protein as your vaccine antigen, because these proteins rearrange, they turn into something like this to allow membranes to fuse and the genome to get in the cell. And so targeting the right shape and surfaces is critical. And so there's other viral envelope viruses with class 1 fusion proteins I'm just listing here. This is RSV, paramyxoviruses like parainfluenza, measles, mumps, etc. Those kinds of viruses also have a fusion protein that does the same type of rearrangement. And so do coronaviruses, the spike protein. And even though they are shaped a little different and have different domains, the way they work is all the same, with this cleavage site in the middle that exposes the fusion peptide. That's what grabs the cell. And then they fold this heptad repeat 1 back to the heptad repeat 2 and pull the virus and host cell membranes together. So the overall strategy for vaccine development for these viruses, we think, is similar. And so as we have been dealing with just this last decade, chikungunya and then MERS, then Ebola in 2014, Zika in 2016, Ebola again, and now this new coronavirus, we've been developing a different way of thinking about pandemic preparedness. And it's based on the entry mechanisms that viruses have. As I just showed you, this is the class 1 fusion protein viruses. Things like chikungunya or Zika would fall into this class of entry mechanisms, and then herpes, pox viruses, rhabdoviruses would be here, and then non-envelope viruses could be a 4th major big category. And we think that we need to address all 26 virus families with prototype pathogen evaluation and vaccine development to address all of the viruses that have a potential for infecting humans and having a pandemic problem. So if you combined pathogen-focused investigators in these four categories and combine that with the core functions that could be organized by other people to focus on the development phase of R&D, then we think that that could be a proactive system for getting ahead of these pandemic threats. As I said, 26 virus families, we think about 30 prototype viruses representing those should be taken through development through at least phase 1 trials. And then there's about 90 other viruses in those families known to infect humans with some potential for increased human-to-human transmission. And we think we should study those over these next 10 or 20 years and develop vaccines that are at least through animal testing. And this is different than the prioritization that WHO or CEPI, the Coalition for Epidemic Preparedness and Innovation, have taken, where they just select some of the most important priorities, but it's not a comprehensive approach. We think this is possible now because of these new technologies. I'm listing here again the structure-based vaccine design, single cell sorting and sequencing capabilities in bioinformatics that allow rapid discovery of human antibodies, definition of antibody lineages that can be new molecular targets for vaccine development, and a single cell analysis of immune responses. These all improve the precision of the vaccine development process. And there's another group of technologies that can improve the speed. And so besides just rapid DNA synthesis, we'll talk about this platform technology where you can manufacture things more rapidly when it's synthetic and where it's always using the same basic chemistry. The reason it's important to go fast is because of the nature of these pandemics. And I'm just showing you a kind of an order of size, the deadliest pandemics in human history. Most of these involve plague caused by Yersinia pestis that can infect people and then become transmitted by aerosol from people. And these are in ancient times mostly, because we now know the pathogenesis or transmission route of this problem, and we can interrupt it, and we can also treat it with antibiotics. There's also two viruses, smallpox, variola, and HIV, that have plagued humans over many, many years. And then there's respiratory viruses. The big one was the 1918 influenza pandemic, which killed upwards of 50 million people on Earth, and maybe around between 650 and 750,000 people in the United States. And then there's these other known pandemics that some of us have lived through, like the Asian flu in the 50s and the Hong Kong flu in the 60s. They killed around 1 million people on Earth. We're already up to over 2.2 million killed by this coronavirus outbreak. So this is already one of the largest pandemics and deadliest pandemics in human history. So we need to go fast for that reason. We also need to go fast because of some of the experience I mentioned earlier in this decade, is that we had a candidate vaccine in the vial on the shelf ready to start a phase 1 trial in the spring of 2014 when Ebola emerged in Western Africa. And despite having that level of readiness, it took until about a year later to start a phase 3 trial in Liberia. And despite that being pretty fast, the outbreak in Liberia waned. It's on this red line. And by the time we started the phase 3 trial, there were not enough cases to get an answer. So we have to be faster than that. We tried again in 2016 with Zika when we made a DNA-based vaccine within about 100 days from the time we selected a sequence. But by the time we got it into a scale up and into an efficacy trial, a phase 2B efficacy trial in Central and South America, it was again about a little over a year. And again, the epidemic waned and we did not get an answer. So another reason to go fast is if you can get your vaccine out during the pandemic and get an answer that can result in licensure, it makes that product available for the future. The other reason is just because this virus is still causing a lot of death and disease. And I just want to make this point because the importance of vaccinating is, I'm trying to illustrate here that eventually, within probably 3 to 5 years, most people on Earth are going to have some level of immunity against this virus. And the question is whether you're going to get there through infection or get there through vaccination. And that's the choice everybody has to make right now. And starting with a non-immune population, you're going to have a lot of people get infected. And the problem is by the time they're immune, you've lost some of the people. And vaccination is a much safer way of getting to a state of immunity without so much death and disease. Another way of thinking about this is the concept of population or herd immunity. In a non-immune population, a lot of people, there's a lot of infection, high transmission rates. In an immune population, you have much lower transmission rates, and that results in outcomes in which there's not only lower transmission, but there's milder disease because the force of infection is less. And there's much less, and in this case, you've got some of the susceptible people surrounded by immune people, some of the infected people surrounded by immune people, which stops the transmission chains. So reaching a state of population or community immunity is really important for the vaccine process. So we have taken the concepts that we learned through RSV, the idea of stabilizing the pre-fusion form of the fusion protein for these envelope viruses. And we've applied that to both paramyxoviruses and coronaviruses over time. And the prototype pathogen for paramyxoviruses for us has been the Nipah virus. And so Nipah is a henipavirus here. And then we've also worked on measles and mumps. And for paramyxoviruses, like RSV, they have this fusion protein. They also have an attachment protein that sometimes has hemagglutinin activity, sometimes not, and is named either HN, H, or G, and different viruses have different properties. Sometimes this is the best target, the attachment protein. Sometimes the fusion protein is the best target for immunity, and you have to determine that experimentally. But all of these paramyxoviruses work about the same way. So over the last few years, we've learned how to stabilize the F protein of Nipah and measles and mumps. And we've shown in all cases that the pre-fusion form of this is much more immunogenic and much more protective than the post-fusion form. And we've also learned that we can make not only the pre-F or just the attachment protein independently, but we can also make these chimeric molecules in which the pre-F is directly attached to 3 molecules of the attachment protein, all in one protein. And so you can either make this with the measles F and a mumps G, or combine both the G and the fusion protein, for instance, for just one virus. We've also applied this to coronaviruses, and coronaviruses are a very large family of viruses that have a lot of zoonotic reservoirs, and they have been coming into the human population, it's thought, for hundreds of years. The alpha viruses, the alpha coronaviruses came in first several hundred years ago. That is, these are viruses that routinely circulate every winter season now. 229E and NL63 are things that we all experience in the winter season. Most of these likely have either had reservoirs in bats or rodents, and then had an intermediate animal host before they came into man. And then sometimes like in SARS or MERS, they could transmit human to human. The first SARS was limited and ended up going away after just a few months. MERS is still a problem in the Middle East, but rarely spreads outside of the Middle East. But now we have this new beta coronavirus, SARS-CoV-2, that has gained traction and has, as we said, infected a lot of people. So because we had just worked on the RSVF and gotten that structure in about 2013, we decided we should work on coronaviruses because motivated by the MERS coronavirus outbreak that was happening in the Middle East during those years. And we had a hard time with the MERS and SARS spike, but eventually started working with one of these endemic coronaviruses, HKU1. We're able to get the spike structure, and it was a very interesting molecule. It's different than RSV. It's extremely mobile. These are the receptor binding domains that have to flip up in order to bind its receptor. And so we finally had a structure of the spike protein of a coronavirus, and then we learned how to stabilize that spike. And in this case, instead of the disulfides and cavity filling mutants, we found that just putting two prolines, substituting two prolines at the top of this central helix between that and the heptad repeat 1 would keep this spring-loaded fusion mechanism from stacking and largely stabilize the molecule. And that had two major effects. One is that it would preserve those highly sensitive apical epitopes on this pre-fusion molecule, like here, but unexpectedly, it also improved expression of the protein. So if you're delivering a vaccine with a gene-based approach like nucleic acid or a vector, you will get as much as 50 times more protein made, for instance, with the stabilized MERS protein than you would with the wild type MERS protein. And we also found that these two prolines were transferable to other beta coronaviruses like MERS and SARS, which allowed us to solve the structures for those spike proteins from those original viruses, and also other viruses like this other endemic beta coronavirus and alpha coronavirus, veterinary coronaviruses, and even pre-emergent coronaviruses that you can find in bats. So we thought that this two proline mutation could be a generalizable approach for coronaviruses. And so we applied that to the MERS, which we chose as the prototype pathogen for the coronavirus family in our idea about prototype pathogen approaches. And we teamed up with Moderna in 2017 because of the work we had done with them on Zika during 2016, finding that their mRNA was more potent than our DNA approach. And so working on both paramyxoviruses and coronaviruses, this is the MERS project, in which we showed that the mRNA delivery at very low doses of 0.1 or 1 could completely protect the animals with this human DPP4 transgenic mouse model, completely protect against disease or weight loss or even viral replication. And even in cases where you had breakthrough infection at this dose of 0.01, you saw some disease, but it was self-limited and not enhancing. So partial immunity led to partial protection. If you were worried about a vaccine-enhanced disease, you would see that green line on this side and with more rapid and severe weight loss. So we thought the mRNA was a good approach that would probably avoid any type of worry about enhanced disease, but also provide a good level of protection. And in December, we were prepared to go forward with Moderna to do the Paramyxovirus phase 1 study with MIPA. But when this outbreak was announced in China, we talked in early January and decided to flip our demonstration project to coronavirus. So coronaviruses, I'm just showing you an image of coronaviruses infecting airway epithelium. And this is a ciliated cell in an airway, and these small little spherical objects are the coronavirus. And you can see that this sphere is decorated with these knobs on the surface, and that is the spike protein, and the spike protein is therefore the major target for most vaccines being developed. And there's over 200 vaccine projects around the world. This is a landscape analysis by WHO, and more than 60 of them have already entered into clinical trials, and at least 4 of these categories of types of vaccines have already been in phase 3 trials, and many of them now have gotten answers back that we can talk about. So the spike protein is the major target. And this is just to remind you that this was not something that we just figured out in early January. This is something that has come from a lot of work over many years. It included the structure-based vaccine design, the thinking about platform technologies and rapid manufacturing, this concept of prototype pathogen preparedness. There's a byproduct of this work that led to human antibody discovery. And then we already had public-private partnerships and academic partnerships in place and prior experience with these public health emergencies of international concern. So all of these things prepared us to react. And so because of the prior coronavirus work and RSV work and preparation work and this experience with not being fast enough, we started on January 10th when the sequences were released onto the internet, and on January 11th ordered the reagents we would need to start a vaccine development program. And very quickly, by January 31st, we were able to be making protein. We had a preliminary spike structure at that time and had a complete structure by around exactly 1 year ago today. And in fact, on the 4th of February, which this is the anniversary of that date, we did our first big set of mouse immunizations, leading to this finding that they were products were immunogenic in mice by February 18th. So within 65 days, we were in phase 1, and within 6 months, we were in a phase 3 trial. And as I mentioned, the byproduct of this is having this protein available allows you to then make probes that can sort B cells from convalescent people. And as we did this with Abcelera and had been doing that for 3 years before, trying to look for cross-reactive coronavirus antibodies, we sent them a probe and we sent them PBMCs. And over the next few weeks, we were able to discover potent neutralizing antibodies. And Lilly made this antibody now as what we called MAB555. They call it bamlanivimab, which is now also approved for emergency use. So starting with a protein, knowing exactly its conformation at the atomic level gives you a lot of confidence that you're working with the right thing. And that allowed us to make diagnostics with the CDC, do this antibody discovery, and then also work with Moderna on the vaccine that we'll talk a little bit more about. And because of this earlier work on how to stabilize the molecule, all of these other groups like Pfizer and Novavax and Janssen are also using this 2-proline stabilizing mutation, which has made their vaccine antigens better. So I'm just going to go over a little bit how mRNA works. This RNA, these single strands of RNA are packaged in a lipid envelope, and that lipid not only stabilizes the mRNA, but helps deliver it to the cell. And the mRNA gets into the cytoplasm, goes through a ribosome to make these proteins. And in our case, we express a protein that's anchored in the membrane, stabilized, and but still cleavable, as I mentioned, that internal cleavage site. And so on cell membranes, it elicits antibody responses that we have shown to be protective. And it wasn't just from this preparedness, but all these other technologies, as I mentioned, have made this possible. The mRNA, in the case of Moderna and Pfizer, is special because it uses not the normal nucleotides, but it uses a 1-methyl-uridine, which that's referred to as a modified nucleotide. So this is a modified nucleotide mRNA approach. And the reason that has been useful is that when you do that to the uridine, it tends to avoid triggering some of these intracellular signals through TLR7 or 3 or RIG-I that trigger interferon production. And when the interferon production is too high and you get too much inflammation, you clear the protein expression and diminish the immune response. So this allows the mRNA to express the protein for a little bit longer and ends up creating better immune responses. And so over these last several months, we've tested this vaccine concept in mice and monkeys and humans and shown that it was immunogenic, even at the upper end of what you would see in convalescent sera. We've shown that it could protect the lungs and nose, upper airways of mice and monkeys. And we've shown that it had a safety profile in terms of its T-cell responses that we thought was favorable with a Th1 bias and avoiding cytokines like IL-4 or 13 that might increase mucus production and make potentially make the disease worse. We've also shown that after 2 doses, the response in older people, 56 to 70, and elderly over 71 is about the same as it is in young adults. And so this vaccine is immunogenic even in the elderly, and it's also Th1 biased in the elderly. And over time, this is the younger adults, the middle column is middle-aged adults, and the right column is the older adults. And these antibody assays are for binding by ELISA, what's called a pseudovirus neutralization, and then 2 other live virus neutralization assays showing that after the peak response after the 2nd dose, even out to 3 months, and now we know out to 6 months, the antibody levels stay relatively constant. So it's maintaining a pretty good antibody response. Better than any products we've tested in the past, especially the protein-based approaches. And so when that phase 3 trial read out in mid-November, the results are shown here. The main result is shown here, where you're looking at an accumulation of infection events in the placebo group over time. And then this is the vaccine group, which had very few events over time. And the main thing to see on this curve is that the protection overall was maintained for at least 3 months, and that some of the protection started after the first dose. And so you see the line separating about 10 to 14 days after the first injection. And the second injection happened here and the lines stayed apart, but you do get some substantial protection even from the first dose of this mRNA approach. And so in the initial FDA advisory committee meeting, the interim data for this Moderna vaccine showed that of the 196 infections, 185 were in the placebo group and only 11 in the vaccine group, for an efficacy of around 94%. And this was true in some of the subgroups or subpopulations as well, that older people had also high level of efficacy. And in communities of color, shown here, that in the 42 infections, 41 were in placebo group and only 1 was in a vaccine group. But all these vaccine infections were mild, and any of the severe disease was all in the placebo group. severe disease, meaning that your oxygen saturation was low, or that you required hospitalization, or that you had a very high respiratory rate. So this was a result that was better than we actually expected. And so when this vaccine related products advisory committee met in December, they voted to approve this vaccine, and unanimously, except for one abstention. And so that's reported here. And on that VRPAC committee was Ovetta Fuller, a virologist from University of Michigan, my good friend James Hildreth, who's president at Meharry Medical College in Nashville. And after that vote, the next day on December 18th, the FDA granted emergency use authorization. And as you would expect, one of the first people to get vaccinated after that was Dr. Fauci. They're getting vaccinated on December 22nd. But since then, a lot of my friends and colleagues and students have been vaccinated with this Moderna vaccine, including Dr. Kizzmekia Corbett, who's a research fellow in my lab, who's done a lot of this work on the coronavirus team over these last 5 or 6 years, and has been central to our work with Moderna to advance the development of this vaccine product. So, and these are some of our nurses, one of my former fellows who's now at University of Maryland, and Dr. Mascola, who's the director of the Vaccine Research Center. So these products are now in circulation. There's more than 32 million people who've been immunized with either Pfizer or Moderna mRNA vaccines. And one of the big questions that people have are, how do you keep watching for safety issues as you go forward if everybody's being vaccinated? And so these are the main ways that safety is still being monitored, in addition to other ways that use these big databases from Medicaid or other insurance companies. This vaccine adverse event reporting system, VAERS, has been in place for many, many years. So this is a place where either citizens or physicians can report things that they think are vaccine-associated adverse events. But for this rollout of the vaccine, the CDC has created another mechanism called vSafe, which is a phone app that prompts you to report any kind of vaccine adverse reaction. And so this is now being used extensively within these 32 million people who've been vaccinated. And you may or may not know, but this vaccine wasn't tested in pregnant women during the trial because they were excluded from the initial 30,000 person clinical trial. But because the rollout was largely in healthcare providers initially, many pregnant women made a choice to take this vaccine as they were pregnant, instead of risking the chance of infection, which is more severe in pregnancy. And so there's now been over 15,000 pregnant women immunized with these vaccines that we're collecting data on. And so far, there's not been any serious side effects that I'm aware of. So in summary, this pandemic response this rapid pandemic response was based largely on prior fundamental basic and translational research that involved both trying to make antigens at an atomic level precision, and also combining that with speed of platform manufacturing and having the pre-established relationships, either with Moderna for the vaccine or with Epsilera for antibody discovery. And I just want to stress too that the technologies we have now and the ways of either making vaccines or analyzing vaccines or understanding immunity is so different than it was even 10 years ago that I think we do have an opportunity to use these technologies to be better prepared for future pandemics. So I just want to thank the PIs that you saw earlier, Jason McClellan, who was at the VRC and worked with me, and Peter Kwong to solve the RSV structure that I mentioned, but has also been very involved in the coronavirus work and structure and engineering. This is my lab, and Dr. Corbett is sort of hiding here, but again, she has led our coronavirus team in our lab to do this work over the last 4 or 5 years with a relatively small group of students. Moderna, our academic collaborators at Vanderbilt, Mark Dennison and Ralph Barrick at UNC, and then all these other groups, especially the extramural division of microbiology and infectious diseases, who helped hold the IND for that phase 1 trial and used their extramural sites to get that done very quickly. So I'll stop there and just show you this very long list of many of the other people at the VRC who have pivoted in their work and have helped and made this whole coronavirus program feasible. So I will stop there, Bill, and take any questions you might have.
Thank you. Thank you so much, Bonnie, for an absolutely excellent seminar presentation. And we'll take questions from live, and also we got some questions in the chat box. But as moderator, I will use my prerogative to ask the first question before they get going into it. Both Pfizer and Moderna are mRNA vaccines, and my understanding they're very similar, but yet they come with different instructions. Like Pfizer, the second dose comes after 21 days, and Moderna's after 28 days. And one has to be really frozen, deep freeze, and -80, and one's just -20. So if they're so similar, what's those differences based on, Bonnie?
Okay, I'm going to tell you the backstory here.
Okay.
The Ugur Sahin, the CEO of BioNTech, which is the mRNA company in Germany that's making the mRNA in collaboration with Pfizer, called me in February 2nd last year and asked what protein I thought he should use in that product. And I told him what we were doing. You know, we're government people who try to share information. And so I told him I thought he should put prolines at position at 986 and 987 to stabilize the spike protein. And so that's what he did. And he started that program. And the day after we started the phase 1 trial on March 16th, Pfizer made a deal with BioNTech to help develop their mRNA vaccine. And so Pfizer didn't start their phase 1 until May, until 2 months later. But Pfizer is big and they have a lot of resources. And so they started their phase 3 at the same time Moderna did in July 27th. So Pfizer helped BioNTech catch up. And one of the ways they did that was by shortening the interval of vaccination. So they went to a 0-3 week to save a week or two and then read it out after a week after that. So instead of reading out the result at 6 weeks, they were reading out the result at 4 weeks. And doing that helped them catch up with Moderna. That's why they got a readout about a week ahead of Moderna in that phase 3 study. And so both groups use mRNA technology that was developed at University of Pennsylvania by Caitlin Carrico and Drew Weissman that modified uridine. Both companies are using that same basic approach. Both companies are using the same basic protein design. And the lipid composition is a little bit different, but it's very similar. And so both of these groups have about the same vaccine. The interval is shorter because they were in a hurry. And the freezing is the temperature for storage is mostly because that's what they had time to verify. The FDA only lets you do what you've been able to prove. And so Moderna had been working on stability and had developed ways of making it so it could be at -20 instead of -70, and had more data on how to store it at 4 degrees for a month instead of just a few hours. So it was really just a matter of what the companies could prove to the FDA was suitable. They're probably also, Pfizer probably could be stored in the same way Moderna is being stored. They just haven't proven it yet.
Gotcha. Okay. So the 28 versus 21 days, that's not really that significant?
I don't think so. In fact, for both products, if you really are just talking about total antibody levels, it's likely that if you made the interval a little bit longer, you would probably get a better boost. So both of these regimens are sort of compressed and rapid because, you know, it's a crisis. We're trying to get answers quicker. But, you know, I think you'd probably find that if you boosted at 2 months, you might even get a higher antibody response than you do at 1 month. Okay.
Okay. Well, we have some questions from the chat box, Bonnie, and I'll start with the first one. Okay. This question says, what is the affinity of the vaccine produced antibody? Is there affinity maturation time with time and later booster doses?
Yes, we have measured, we've tried to measure the avidity of polyclonal antibody responses in the vaccine, and we know that it does increase over time. So the other thing we know is that after the first dose, you don't see a lot of neutralizing activity. You see a decent antibody response for binding activity, but not a lot of neutralizing activity. You don't really see a lot of neutralizing activity until after the second dose, which probably means you're getting additional somatic mutation and better antibody affinity being established. And so it's an interesting finding because as you remember, I told you it looks like there's pretty good protection after the first dose. Mm-hmm. And that's before you can measure a lot of neutralizing activity in serum. And so we don't really entirely know. This protection looks like it's very correlated with antibody responses, but as I mentioned, you can see pretty good protection before you get much neutralization.
Gotcha. And we have another question here, Bonnie. This person says, what is the possibility of having been vaccinated and if you're a silent carrier with no symptoms and get vaccinated, is that, what does that do to the spread or how's that impacted in some way?
Well, you know, it's usually in infectious diseases, you get better immunity from an infection than you do from a vaccination. In this case, it looks like the vaccine immunity is actually stronger than the infection immunity. If you compare people who get infected in their convalescent sera, unless they have very, very severe disease, they usually don't get as high of antibody response as the vaccines are inducing. And the protection against reinfection is stronger with the vaccines than it is with infection. And so that's been a very curious thing. And so when people are infected, it's still recommended that they get a vaccination. And the recommendation right now is that they wait for about 90 days, partly because if you wait a little longer, you'll get a better boost. You need to let your memory B cells kind of calm down into a resting state before you boost them. But it is recommended that infected people get boosted. Now, if you get the vaccine and you're an asymptomatic carrier, I don't think there's evidence or reason to think that that would hurt you. And so I think it's okay if you do that. And but there's also evidence that I didn't show you that maybe this vaccine even protects against asymptomatic carriage because at the time of the second dose of Moderna, they measured, they did PCR testing of the nose. And of all those people, there were about 42, I think, people, 52 people who had positive PCR out of those 30,000. And 38 of those were in the placebos and 14 were in the vaccine group. So it suggests that maybe, and they were asymptomatic, it suggests that maybe this vaccine is even protecting against asymptomatic carriage. And we know in animals that it does reduce the amount of virus in the nose, which probably means it's reducing transmission. It may not fully protect your nose, but it probably reduces the virus in the nose.
Okay. And we have another question here. It says, what is the potential that there could be side effects a year to 5 years out? Or how effective is the vaccination produced antibody in reducing or eliminating the viral load? Is natural infection more effective in producing high antibody than the vaccine? I guess you answered that already, but the other two.
Right. Okay, well, most vaccine side effects historically, 99% of them happen within 6 weeks after vaccination. It's rare to see anything related to vaccine coming farther out than that. And that's one of the reasons the FDA used that 2-month cutoff, is say you have to have at least 2 months of safety data before we'll look at this for emergency use. And so the other thing is the mRNA is probably the most elemental, simplest approach for generating a protein. It is, it can only be detected for 24 to 72 hours or so after injection, and then it's eliminated. No, you can't detect it after that. And even the protein that it makes is there not more than about 3 to 7 days. And so this is a very simple, elemental way of making a protein to create an immune response. And it's much less of an inflammatory event, or much less of a, you don't get all the other parts of the virus. This is only about 10% of the virus that you would get during an infection. So we obviously haven't followed people for 5 or 10 years yet, but people have been doing mRNA immunizations for 10 years, and there hasn't been any evidence that these kind of long-term side effects would happen. And I think it's much more likely to happen with the virus itself, to cause these kind of long-term things that people are worried about.
Okay, this next person says, first, thank you for your great work and presentation. Then they ask, how long do you expect the immunity to last?
Right. Well, first of all, thank you. I paid that person to say that. So anyway, you know, we don't know yet how long it's going to last, but it's encouraging to see these antibody curves being fairly flat. And so we're hoping it would last more than a year, but this is something we're going to have to test and see. The other thing that might not make it last as long as we wanted is because you've probably heard of the variants that have been coming up. There's a UK variant that people talk about, and now one from South Africa, one from Brazil, California variant. There's going to be a lot of other variants that have very complicated numerical nomenclature that I can barely remember. But as these variants come up, you know, every time the virus infects a new person, it has a new chance to adapt. And so these variants, if we don't get the virus replication and spread down, we're going to still keep getting new variants as the virus keeps adapting. And so it may be that eventually these things will adapt enough that we'll have to change the vaccine. And these types of vaccines are not that hard to change. It's sort of like a plug and play type of system that you can just put in a new sequence. But it is one of the reasons why getting vaccine deployed more quickly and thinking about this in a global way to immunize everybody on Earth so that we don't have these pockets of virus adapting, and to be still continue to be careful with physical distancing and masks. It's one of the reasons why we have to keep doing that, because we don't want these viruses to adapt.
Okay. The next question here, Bonnie, is that can you please explain why some people report really bad side effects from receiving the second dose of vaccine relative to the first dose?
Right. It's interesting because in the mRNA, the side effects are a little worse after the second dose. And that is true. Both the local and the systemic side effects are worse after the second dose. So the type of side effects you get are arm pain at the injection site, sometimes a little bit of swelling, and sometimes a little bit of redness. On the second dose, you might get a little bit of fever, muscle aches or tiredness, fatigue that last usually about a day or maybe 36 hours, and then it goes away. So those symptoms are more severe, and maybe 10% of people end up needing to take Tylenol or Motrin or something like that for the symptoms. But overall, it's not that different than many of our other vaccines. And that response, that reaction is evidence that you're making an immune response, which is exactly what we want you to do. And again, it is not the best day. That's the day after the second dose, but it's a lot better than getting the infection.
Yeah, I can imagine that. We got a few more questions, Bonnie. You okay?
I'm good. Yeah.
Okay. This next question is there evidence about how effective antibodies from vaccination with the Moderna vaccine are against the virus with the E484K mutation?
Right. Yeah, that's a good question, because the glutamate to lysine mutation at 484 is the one that came up in the South African variant and also in the Brazilian variant. And that mutation did change the overall level of neutralizing activity from vaccine sera against those viruses. And some of those reports are being posted now in bioarchives, and some of them are starting to be published in peer-reviewed journals. But that mutation does change the overall sensitivity of the virus to neutralization. But we now have data from the Novavax protein study and the J&J Ad26 study in South Africa, where most of the virus infections in those studies was from that South African strain that has the 484 mutation. And even though the protection against mild disease or moderate disease, the symptomatic coronavirus disease, was diminished maybe by 20% or so, there was still no severe disease. There was nobody in the vaccine group got severe disease. And so we think that there's enough buffer in these vaccines with 95% protection and a complete protection against severe disease that it will still cover this current set of strains. The question is, as these mutations accumulate, if we get another set of mutations beyond 484, will it still be okay? And so Moderna and Novavax, at least, have already announced that they're making new vaccines with these new strains, sequences. And so, there'll be a system to track and keep up with these strains if we need to change the vaccine.
And this question is asked, did you test CTL response, vaccine stimulated Th1 type of response?
Right, we did test both CD4 and CD8 responses, and they're reported in some of those phase 1 clinic results. And you don't detect a lot of CD8s in the blood from the mRNA vaccine, but we think that they're still there. For the gene-based vectors like Pfizer or Moderna mRNA or the J&J or AstraZeneca ad vectors, we think that CD8 T cells are being induced. You're not likely to get CD8s from the proteins like Novavax, but we think that those are probably useful and helpful, especially in clearing virus from the nose. And so one of the reasons we chose the mRNA approach is because it would give you some CD8 T cells.
Okay, this question asks, what precludes the use for other infectious diseases? For example, why is it not used for HIV or CMV and so forth? I guess they're talking about the mRNA platform there.
Well, you know, there's just never been enough incentive or commercial interest or investment to ever get the scale up finished. And so now, because of all the investment in this year, mRNA is now scaled up. And so I know that Moderna has very effective products and early phase data on a CMV vaccine approach where they deliver 6 different genes, the pentameric piece and the GB for their CMV vaccine. And so I'm expecting that there probably will be other mRNA vaccines coming out after we get past this pandemic problem.
Okay, and this next question, Bonnie, I'm sure this one is dear to your heart. It says, how would you convince those who are hesitant to take the vaccine due to historical or unethical practices in medicine, particularly toward communities of color, to take this vaccine?
That is the question of the day, of the year, of the hour. And there are a lot of reasons, historical reasons, to have distrust, and there's even current, immediate, yesterday reasons to have distrust. But we still have systemic racism and implicit bias even among our healthcare providers, who often make decisions that are biased against one group or another. And so it is a very hard thing to ask people to trust. But I think, and you know, the other problem is you can't gain trust quickly. Trust is something that takes a long-term relationship. And so it's very hard to convince some people that they should trust this time and trust the vaccine to work. And the only thing I can say is that I just ask people to look at the data and just look at the information and make your own judgment. Don't trust the systems, but just trust your own evaluation of the data that's being published. And, you know, I don't know what else to say, but my daughter was a volunteer in the Moderna vaccine study, and she's now been immunized. Trying to get my wife immunized, and I trust the vaccine safety enough to have my loved ones immunized myself. And so I can say that. I can also say that it's not just hesitancy. And hesitancy might even be used as an excuse for maldistribution. What I'm equally worried about, or maybe more worried about, is that this vaccine won't be accessible to people of color in the same way it is to white populations. It's because the pharmacies may not be as close, but it's also just because of vaccine allocation and decisions being made at local levels about who should be prioritized within a hospital system, for instance. And I think there's already evidence that even among people, there's a lot of Black people who would like to be vaccinated and they cannot get the vaccine yet.
Yes.
So hesitancy is not the problem yet. The problem right now is maldistribution, and there is a lot fewer percent of minority populations being vaccinated than white populations. So I worry about access and distribution as much as I worry about hesitancy.
Thank you. We only got a couple of questions, Bonnie. Is that okay?
I'm okay. I think, let me check.
I know we're working you pretty hard here.
It's okay. I really, you know, part of the problem with hesitancy is that you can't really explain these things in bullet points on a TV evening news show. You have to have a conversation. And so I'm actually very happy to be able to have a conversation.
We really appreciate it. We really do. And this next question says, For a person who is already infected once with COVID-19, does the first dose itself serve as a booster dose, or is the second dose required for such a person?
I think it does serve as a booster dose. You will get boosted if you get a vaccine after you've been infected. And so the problem is we don't know enough yet whether that one dose is enough and whether it will last and be durable and sufficient, or whether you need that second dose. You also, if you're immunized with one dose of vaccine and get infected, like I said, it's partially protective. You get boosted by that as well. So the infection can boost the vaccine, and the vaccine can boost the infection.
Okay, and the next question is, natural immunity matures to high affinity antibodies as the virus antigen clears. How does the vaccine present lower concentrations of antigen with subsequent doses since the antigen is generated in situ?
Right. The mRNA goes into some of our muscle cells because that's where it's injected, but it's also going into some of the dendritic cells and macrophages, and some of the particles even apparently go through the lymph and get into the subcapsular sinus macrophages. So the vaccine is being presented in places that go directly to draining lymph nodes or directly into draining lymph nodes. And so the other thing that's happening with the vaccine is it's getting a very strong T follicular helper cell response, probably better than what you get in natural infection because of all the regulatory things happening in infection, and some of the genes that the virus uses to avoid or evade innate immunity. So it inhibits type 1 interferons, it does all these things. And so for whatever reason, we are getting better immune responses from the vaccine that is in the absence of all those other viral proteins than we get with the virus. And we think it's in part because of such effective induction of TFH cells and production of IL-21.
And this is the last question, one right here. What do you think about some companies trying to evaluate the benefit of getting a third dose?
Right. Well, this is going to continue to come up, and most companies are thinking about how to boost. Moderna is going to have a group that gets a boost at 6 or 12 months, for instance. AstraZeneca is making deals with Pfizer to boost their adenovector with mRNA. I think the similar thing is being done with mRNA priming or advector priming and a Novavax protein boost. So I think a lot of these different things are going to be tested, and then you can watch and measure the durability of the antibody response and the level of protection. So right now, I think it's largely just scientific experimentation. I don't know if it's going to be needed in the future, but a lot of those kind of studies and questions are being asked.
Bonnie, I want to thank you, and I wish you could see all the positive comments you're getting in the chat box, but you get a lot of them. And I thank you for a great seminar, and the information was so contemporarily pertinent, and it's just been wonderful experience for us, and we thank you for that. And I also would like to just say that I think, and as near as I can tell, this seminar was probably the most attended seminar we've had, at least in a long, long time. So thank you very much, Bonnie. And I think we worked you hard enough today.
Well, thank you, Bill. It was really a pleasure to be with you today, and nothing could make me happier than to know that somebody decided today that maybe I will get vaccinated instead of waiting to be infected. Yeah. Because that I think is what we need to have happen.
Yeah. So once again, thank you so much. You know, at the very beginning, I was talking with my wife, and I said, "No, I want to get Moderna vaccine. That's the one Bonnie worked on. It turns out that in the situation we got in, we got the Pfizer. I said, okay, let's get the Pfizer. Can't we?
No problem. And let me just say that if an FDA has given approval to a vaccine, and if you can get your hands on it, I would take it. I would take anything as soon as you can that the FDA has approved. Because if they've approved it, I think it's going to be relatively safe and much safer than an infection. And all of these vaccines to different levels of effectiveness, but they all seem to be effective against severe disease. So I would encourage people to take whatever one they can get first.
Yeah, yeah. The one we had access to was Pfizer. I said, man, I want the Moderna.
That's okay. They still used our protein.
Okay. Bonnie, thank you so much. I really appreciate it.
All right.
And you have a great day now.
All right. Thank
Bye-bye.
you.
Bye-bye. And thanks everybody for coming. We appreciate you coming. We appreciate your questions. Thanks so much. Everybody, let's